So let's go back to the nucleotide basis, and we call it as, following their names, Watson-Crick base pairing. So the nucleotides composed of 4 different kinds: adenine, guanine, cytosine, thymine. Or in RNA case, instead of using thymine, we use a uracil. Okay, so you can see this pentose, phosphate, and DNA base. So this A and C, T, G is a building block monomer of nucleic acid, and that's a nucleotide. And they polymerize into a linear sequence of this A, T, G, C, T, T. And this has, it's called single strand nucleic acid. And while DNA is in fact a double-stranded in most cases. So you see found the another one, but when they do interact, you can see A always interact with the T, T goes to A, and G, guanine, goes to C, and C goes to G. So this is a complementary matching, and the secret inside is the hydrogen bonding for each to be matched. So when they interact, they form a helix structure, double helix structure, and we call this a hybridization. And this single nucleic acid chain can combine with another. So complementary means A always go to G, T always go to C, okay, vice versa, and to form a double helix. And this process of 2 single strands of DNA assembling into a double-stranded DNA is called hybridization, and in molecular biology, DNA hybridization, when these 2 strands have complementary sequence. So that's very important. What it means is, if we know the information of 1 strand of DNA that's coming from parents, and you readily can make another one which is complementary, then that information is identical. Now, these DNA, the genetic information can pass from one to another, and that's the secret of our life. So let's go down a little more. That we mentioned about purine and pyrimidine from Watson and Crick's Nature paper. And what that means is this DNA basis, if you look at that one, the G and A, guanine and adenine actually have these 2 ring structures, while thymine and cytosine has this 1 ring structure. And in fact, they have this dotted line, it's a hydrogen bonding. So this is why G goes to C and A interact with T specifically for DNA. How about RNA? RNA, the nucleotides are almost the same, except this thymine is replaced with a uracil, which is also similar shape. So it's similar. So Watson and Crick base pairing rule tells if we have 1 DNA strand and the complementary DNA strand will be matching in this way. While, you know, for producing a protein, we use DNA as a blueprint to produce RNA and protein. So for that process, we need to have a DNA as a template. And then we have this, create this RNA which is complementary. You can see A goes to not T because RNA uses uracil, AU, CG, CG, AT. Actually, I think this is a mistake. This is supposed to be U and AU and CG. Okay, so these a single structure called pyrimidine is adenine and guanine is called purines, and this cytosine and thymine is called pyrimidine or uracil, single ring structure. And this hydrogen bonding capability of these bases, in fact, determines these base pairing rules. And, you know, as you can see here, 2 and 3 hydrogen bonding. So this force holds the complementary base pairing match between 2 DNA strands in a very stable double helix. And that's hydrogen bonding. So now, first part of explaining, so why is this so important? And there's a very famous dogma of molecular biology proposed by Francis Crick. So what is dogma? Dogma, we only hear from maybe religion. So it's like principle or set of principles laid down by an authority as incontrovertibly true. So it's a little weird to use dogma here, but Crick used it because he observed this phenomena. And our fundamental question, and in fact, the molecular machine in our body as a functional unit is protein. Then how this blueprint of DNA, in fact, gives out the protein, and that's where the information goes. So we think it as the information flow, and how we think about this information goes from one side to the other side. And that's summarized in this picture. So from DNA, we use this as template, and that information we transcribe. It's a copying into the RNA as one-to-one. So from DNA, we transcribe it into RNA. And this RNA molecule now gives rise to protein. And that process, you remember, RNA has these 4 monomers, AUGC, these 4 monomers, and this AUGC, blah, 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 this sequence somehow gives out the protein. And remember, the protein is polypeptide chain composed of amino acid sequence, which has about 20 different kinds. And so we have to translate this RNA sequence into a protein amino acid sequence. So that's why we named it as a translate. And who does this job? Is also, that is a protein, and using that is especially enzymes. And these enzymes called RNA polymerase. And DNA has to copy self-replication, such as one single cell containing DNA, divide and produce another one. They have to copy exactly the same genetic information. So DNA has to replicate itself, and that we call DNA polymerase. And the information comes from DNA to RNA, and RNA to protein. This unidirectional information flow is recognized by Francis Crick, and he named it as central dogma of molecular biology. So the process of translation and transcription that produce messenger RNA and proteins require coordinated chemical reactions, which are catalyzed by enzymes called DNA and RNA. These enzymes make a polymer from this unit, so polymerase. You recognize a lot of these A's as actually naming usually enzyme. So in a nutshell, protein is a biochemical compound made one or more polypeptide chains, which perform functions in cells and organisms. And the process of information transfer from DNA to RNA is called translation. And the sequence, because the sequence of nucleotide from DNA is one-to-one matching into another nucleic acid. And that of the RNA we call transcription. And one kind of an RNA which is involved in making protein is called the messenger RNA. And to protein is information of this mRNA into a linear sequence of amino acids that require translation of mRNA sequence that will become a protein. It's called translation. So the first paper who proposed this is Crick in Nature and Central Dogma of Molecular Biology. You can take a look at this, and we will discuss a little bit of review of genetics to move on more deeper meanings of and processes of Central Dogma. Thank you for your attention.